All right, can everyone hear me? It's a big stage, wow. Thanks for sticking around, thanks Ken. Might want to throw one of those bean bags over here in case I fall off, cushion my fall.
Hold on, Jim.
Little nervous.
Technical difficulty. I pulled it out.
We good?
Yep.
All right. So in all of our businesses, we track or want to track lots of things. And so your list probably looks different than this, but the concept is the same. So we need to track these things in order to improve them and to innovate. And in our experience, there's lots of tools to help us track these things, some better than others, and some work really well for specific things. But often the weight of these tools bog users down. Many are inflexible and costly, but we've found JIRA to be the perfect fit for our tracking needs. It's super flexible, enough to apply to a wide range of business processes, and it's really intuitive. So our users quickly grasp the concepts, and we've seen usage and productivity explode. So for us, JIRA has become indispensable for the things we're trying to track and is now embedded in the culture of our workplace. And that allows us to tackle sort of bigger and bigger projects. So I'm Jim. This is John Stalker here, my good friend and colleague. And we'd like to talk to you today about the Broad Institute's use of JIRA, how we use it to track things in a genome sequencing lab. And when I say things, I mean almost everything. And although the tools we've built act specifically in a genome sequencing lab, the concepts can be broadly applied to any business process to increase efficiency and foster collaboration. So if it'll work for something as esoteric as genome sequencing, it'll work to improve your processes as well. In fact, John and I have yet to come across a process that isn't a natural fit for JIRA. So the Broad Institute is a nonprofit collaborative research organization. It was founded in 2004 by Eli and Edith Broad, who are amazing philanthropists and just amazing people, along with MIT and Harvard University. There are also affiliations with many of Boston's top research hospitals, and the goal here is to transform medicine and health by applying collaborative science to the biggest problems facing biotech and medicine today. And this is done through collaborations with over 100 different private and public institutions in more than 40 countries worldwide. So the Broad is organized around scientific programs on the left here. These folks are interested in answering challenging questions about relevant problems in the biomedical space. And on the other side are platforms arranged around technical capabilities. So this creates some interesting collaborations where a program that is studying a particular disease like cancer or diabetes can call on the technical capabilities of several platforms to help them answer that particular problem. So John and I work in the genome sequencing platform and have applied JIRA to many of our processes and collaborations over the past several years. But you can already see the potential for applying JIRA to the larger organization. Just want to give you a little background on the history of genome sequencing. So here I'm plotting the cost of sequencing a human genome, these blue dots, over time since 2001. So this is compared against Moore's law, and I'm interpreting Moore's law here to mean if the cost of computing were to decrease by half every 2 years. In this case, we're looking at the cost of genome sequencing. And that's the red line. So this scale here is in US dollars, and it's scaled logarithmically. So the original human genome project, the effort to sequence all 3 billion letters of DNA that make up your genome, took over 10 years and $3 billion to complete. So that project completed in 2001. And that was big science, with dozens of labs from around the world contributing data and collaborating on technology development. And since then, we've seen an advancement in technology that has incrementally drove down the cost of sequencing. So from the advent of automated sequencers and automated sample prep that allowed us to do several genomes per year, to the more advanced so-called next-generation sequencing instruments, which in a highly parallel fashion could sequence a genome in a couple months, or just a couple weeks for those. Sorry, John. The current state-of-the-art sequencers that we have today can generate enough sequence data to complete your genome in a couple days for a few $1,000s. And the goal here is to drive the cost of sequencing below $1,000 so that it becomes a routine diagnostic that clinicians could order up much like a CAT scan or other standard test. And this curve is impressive in that there are few technologies that can surpass Moore's law, few things that are evolving as fast as genome sequencing. The problem with that, though, is now by outpacing Moore's law, you switch the bottleneck to computing. And so we're approaching a place where we can generate sequence data faster than we can analyze it. So this reduction in the cost of sequencing has allowed us to contemplate sequencing more and more samples, more genomes. So the orange line here, plotted against the right axis, shows the cumulative number of samples sequenced at the Broad over the same time frame. So from that single sample, that first human genome in 2001, to roughly 1,000 samples per week. And this is important because to start to understand the genetic underpinnings of complex diseases such as cancer, heart disease, and diabetes, we need to be able to compare the genomes of thousands and potentially tens of thousands of individuals. So with the advent of new sequencing technologies, these large projects could begin, and we begin to see an influx of samples, all of them needing to be tracked through our lab. So standing at the bottom of this cliff here and looking up was really this oh shit moment for us. And JIRA had been living under our noses for several years. We've been using it in more traditional ways for bug tracking and software development. When thinking about what it would take to track thousands of samples per week, I mean, we looked at some other solutions, and then John had an epiphany. Hey, we can use JIRA for this. So we started using JIRA in this way in May of 2010, at the start of this curve, looking up. And this project, LC Sets, which John will describe in a minute, was designed to help the lab deal with the increased management load that these numbers present. And we haven't looked back. All right, here's just a little introduction to genome sequencing. How do we do what we're doing here? So each cell in your body contains DNA. So packed into chromosomes within the nucleus, this is the so-called code of life. In each cell, you have 23 pairs of chromosomes. So we inherit one of each pair from our mom and one from our pops. And yes, that's actually my mom and my dad. But no, that's not me. I'm actually a lot bigger than that, and I no longer fit in my parents' laps, even though I often still try. That's my son, Finn. And if we look at these chromosomes under the microscope, we discover something amazing. JIRA is in our DNA. John made me keep that one in there. I had to get rid of it. So actually those chromosomes are made up of long stretches of DNA, two intertwined molecules, this double helix that was famously described in 1953 by Watson and Crick. And this is the molecule which carries the instructions for life. So those instructions are encoded by 4 bases: adenine, thymine, guanine, cytosine. And they base pair with each other along this spiral ladder, A with T, G with C. We can take this DNA then and place it in a DNA sequencer. So shown here is the Illumina HiSeq. It's roughly a $700,000 instrument. But it essentially works like the BAT computer, right? I could stick DNA in and I get a sequence of digital bases or letters out. And there's a lot of letters here, over 3 billion in the human genome. And what we're looking for are mutations, those places in the genome where a letter is different from some reference sequence. And so for cancer, we want to compare the letters in your normal cells to the letters in a tumor cell. And we want to discover the mutations then that are driving that cancer. So right there, there's a T in the normal and an A in the cancer cell. And the hope is that with a molecular understanding of a particular cancer, clinicians could offer personalized care that is better tailored to a patient's prognosis based on a spectrum of mutations present in that cancer. So now that I've shown you how to sequence a single genome, John's going to take it to the next level. He's going to show you how to use JIRA, how JIRA plays a role in our ability to sequence thousands of genomes per week. Here you go, John.
All right, thanks, Professor Jim. That was great. Everybody liked Jim's science lecture there. It was wonderful. All right, so through this segment, we're going to take a look at 3 JIRA projects in use in our lab that are tracking physical things. These are the thousands of samples Jim was talking about, as well as the automated instruments that are doing the bulk of the work and our sequencing runs. So first, I'd like to introduce you to the archetypical laboratory process, beginning with the players. These are the folk in the lab who will be collaborating over the lifespan of an entire genome sequencing effort. So first, as Jim described, we have our scientists. These are the research partners that have specific scientific questions to be answered through DNA sequencing. They'll collect biological samples related to their specific area of research. Now, besides the mundane, typical sequencing projects like cancer, HIV, and deadly European E. coli, we've also got some really cool collaborations on the go with groups like the University of Alaska Fairbanks, where we are sequencing microbes collected from ice cores drilled from miles below the surface. It's pretty cool stuff. So all these samples that we receive are tracked through various barcoding mechanisms. Like pictured here, we have tubes that have 2D data matrix barcodes on the bottom. We have millions of these tubes. So the samples are gathered up in batches, racks of tubes, and sent to our project managers. Now, PMs manage the communication between the scientific collaborators and the lab, matching scientific needs with technical capabilities. And here, at long last, is JIRA. So just put it in the back of your mind for now, but from here on out, JIRA is a way of life for us. The task of processing samples and preparing them for sequencing is handled by various lab personnel. So coordinators manage the day-to-day effort in the lab, ensuring that sample batches are on track, while the technicians are the fine folk that get the real work done. They do a fair bit of this. This is pipetting, so moving samples from tube to tube, reagents, plate to plate, but they actually do a fair bit more of this. This is running the automated instruments that we have in the lab. And now these machines are what really enable us to do the science at this level. So we have machines that cut up DNA into really small bits on the left there, machines in the middle that do bulk pipetting operations, and on the right, actual sequencers. But for all intents and purposes, they're all just robots. Now, all these lab resources are organized into functional units or teams, and JIRA helps track the handoff from team to team and group to group. Now, JIRA really facilitates communication between all the players through this entire process and naturally promotes a highly collaborative team environment, even when the roles and responsibilities are so diverse. The ultimate goal, though, as Jim described, is to produce sequence data that is consumed by analysts and bioinformatics computational biologists. Now, if we rotate this process vertically, we arrive at something that starts to look a little familiar. Here's a screenshot of the workflow designer in JIRA. Our library construction set, or LC set, JIRA project, has workflows that look a lot like this. States here represent the various functional units and teams, buffer points throughout the entire process. An LC set issue represents a batch of samples traveling through the lab together. Organizing our workflow this way allows us to monitor a wide range of performance factors. We can quickly see how many sets are in each group. You just go to the JIRA default dashboard to see that. We can monitor cycle times. How long has an LC set been in flight or in a particular group? You just go to the Transitions tab in any of your issues. And in reality, things aren't quite this simple. We have many issue types representing the different types of LC sets in the lab, each with its own complex workflow. But the point here is that you can do this in JIRA, and it's really not that hard. So workflows are obviously only half the story. We also make heavy use of custom fields. we try to limit ourselves to only the most important data from a process tracking and project management standpoint. Most of the low-level metrics related to process are better stored in a laboratory information management system, or LIMS for short. In JIRA, though, we actually capture data that makes it easy to link from JIRA to and from the LIMS through custom fields for project ID or sample ID. So here it all comes together in our LC Facebook dashboard. At a quick glance, you can see how many batches are with each team in the process. That's on the left there. On the right, you can see, things are actually moving pretty smoothly through the lab, nice linear slope. Now, even though pie charts are pure evil and should never be used, we get a quick glance at what kind of samples are moving through the lab here. Now, if we scroll down, you'll see where LC Facebook gets its name with this mile-long activity stream. If we go and take a quick look at one of these issues, a Hybe Cell LC set, you can see the custom fields in use, like which version of a protocol is in use, which sequencing instruments are used. GSSR IDs are our sample IDs that link to the LIMS, QC metrics, and all of our transition timings for cycle monitoring. In short, you can take any business process, even something that's far removed from typical, like genome sequencing, and boil it down to a workflow. So now let's take a closer look at the final step in this particular process. Samples enter the sequencing group to be turned into sequencing experiment. We have multiple workstations at each step to provide parallel throughput and redundancy, which is good because sometimes when you go to do something, robots burst into flames. All right, well, they don't always burst into flames. They never burst into flames, but they do break down on occasion. But we have a JIRA project for this. Our Crazy Robots project is used to track the equipment maintenance requests. So when an instrument is in need of some TLC, an operator can create a ticket, or in some cases, the instrument can create a ticket automatically using the REST APIs in JIRA 5. Now, if an operator comes along forgetting or not knowing that a robot is in distress and has an open service request and attempts to use that instrument, the robot queries JIRA always as its first step in its execution plan, determines that there are open issues, and prevents that protocol from running. So in this way, JIRA is actually controlling physical process in the lab. It's pretty cool. Not until a service technician has come along, fixed the robot, and closed out the issue will JIRA allow samples to be processed at this station. So now if we move back to the healthy workflow here, the final step before being ready to sequence using Illumina technology, our current workhorse platform, is to load the samples onto something called a flow cell, pictured here. Now the robot that performs this operation will create a ticket for each flow cell that it processes, again using the REST APIs. Now in our flow cell tracking JIRA project, an issue represents a flow cell. So this is, again, it's very similar to the LC set project, where the issue represents some physical unit of work traveling through the lab. When the flow cell has been prepared, it is finally ready and loaded on a sequencer. Here, the sequencer takes an active role in the JIRA workflow by transitioning the tracking issue into the sequencing run started state and filling in some custom fields, like the name of the sequencer that this flow cell got loaded on. So, we've heard this a lot this week, but again, you can integrate and automate your business processes with JIRA using RESTful web services. So here's a quick look at our flow cell tracking workflow. When a run is started, we have several loopback transitions to the same state that we use to capture timings and gather some control metrics through the course of a run. And in the event of a failure, we capture troubleshooting metrics. Now here's an example of one of these flow cell tracking issues with a variety of custom fields. We have information ranging from which instruments were used to prep and sequence the instrument, what, if any, fail modes were encountered along the way. We've got QC reports attached, or PDF reports. The usual comment stream, which we love because from start to finish, downstream operators can see any sort of anomaly that happened along the way in the course of creating and sequencing this flow cell. And the usual transition timings at the bottom. Now, JIRA is great by itself, but it's even better when you realize just how much data you really have access to. All your custom data, state transitions and timings, comments, it's all data-based. The power of JIRA becomes limitless, though, when you mix in the idiot savant brain of Jim Meldrum here. By bolting data exploration and visualization tools, such as Tableau, pictured here, on top of our JIRA database, Jim has taken JIRA beyond workflows and issues and into the world of data gathering for statistics and analysis. Tableau, for anybody who's not familiar with it, is data visualization software that can tie together multiple data sources from databases, Excel files, CSVs, and makes it really easy to explore complex relationships across all those data. At any rate, what you're seeing here is a Tableau report that ties together the custom fields we saw in the flow cell tracking project and native JIRA data, like issue creation date. Here we can see trends for particular fail modes over times and the computed ratio of successes and failures. Again, all this data comes from JIRA and JIRA alone, so it's really great. So, now that we've taken a close look at some projects to track physical things in the lab, Jim is going to close it out by describing our fourth and final JIRA project and why it is better than paper. Bring it home, Jim.
All right. Thanks, John.
Oops, I moved on.
So in any typical lab or office, there are lots of people independently accumulating knowledge. This activity gathers in file systems, databases, or worse, spreadsheets, emails. And in most labs, the classic repository for this type of information is the lab notebook. So the problem is that we have all this knowledge squirreled away in these lab notebooks, and this makes collaboration nearly impossible. An article in the journal Nature in 2005 looked at the current state of electronic-based lab notebooks across the physical sciences. And the article seemed to herald the imminent arrival of the eLab notebook, but stated some of the challenges. The experimental protocols are continually changing. Redesigning the pages of e-notebooks and adding fields and tables usually entails modifying the structure of the underlying database. It went on to say that this then requires the skills of database engineers, a luxury few labs enjoy. So this article obviously hadn't looked at JIRA. I mean, we don't need skilled database administrators. JIRA adapts to your changing needs, essentially meeting this challenge. I mean, I couldn't even code my way off of this stage here, but you know, I can configure JIRA and derive real value from these workflows and fields. So fast forward to 2012, and a follow-up article then revisited the state of the eLab notebook. So there are lots of players in this space, lots of different software companies, but adoption is still slow, and some of the old challenges remain. The greatest challenge is not to create a tool that can do everything, but one that is fundamentally easy to use. And I would argue that JIRA is easy to use, and it also allows you to do, well, maybe not everything, but almost everything. So challenge met. Okay, so how does this play out using JIRA? So everyone has their lab notebooks, and my dream looks like this. So we're going to gather up all these paper-based notebooks into a great big pile, and we're going to set them on fire. We break a record for flames in a presentation.
Well, what happened to that guy?
Oh, he must have got a little too close. He had a clipboard or something, so he probably deserved it. But then this is my goal. I mean, to move this information out of those squirreled away lab notebooks and get them out in electronic format where they can be shared. So, right, it's sort of obvious. But it doesn't need to be iPads. I mean, we have computers throughout the lab, but JIRA is the perfect place to capture this information. So let's focus in on an example. We're constantly trying to improve our processes, make them faster, cheaper, or more reliable. And this happens in many ways, and we've tried to map these activities to JIRA issues and workflows. For example, there are just classic projects that are initiated from various places in the organization. So this boss man here walks up and declares, hey, we need a better shearing protocol. So we use JIRA story issues here. Probably, I mean, essentially it works like a project for us. We probably even should call it issue type project, but. And so then this story spurs activity in the lab, right? A technician then may go off and perform an experiment, tweak something with the robots, alter a protocol, maybe add a new technology to the process to see if they can make an improvement. We also captured this in JIRA using a custom issue type experiment. So the results of this experiment might give birth to an idea from elsewhere in the organization. And I got too many meeples running around the screen. So we also, we gather up all these ideas. We can use JIRA to sort of vote on them, and even then the best ideas can graduate into other experiments or projects. And then we also just sort of have mundane tasks. So you can see here, we just need to get this file over to John's office, so we can gather up just those plain old tasks as well. And so this forms the basis for the main issue types we deal with. So we use the story or project issue type as an umbrella for the other issue types. And we were able to create a hierarchy using a simple custom link with a parent-child relationship. And then this gives us limitless levels in that hierarchy to play with. And managers can organize these relationships in ways that suit them. So we recently installed the structures plugin from ALM Works. And we were playing with that to help us manage these sort of ad hoc relationships. So we have some additional issue types that we use. We also have a document issue type. So we're sort of disappointed with JIRA's ability to annotate or describe plain old attachments. So by adding a document issue type, we then are capable of describing those documents in more detail so people can easily find them and learn from them more efficiently. We also have a process change here, and this is simply a note with a timestamp to let people know when something in the lab has changed. And then I can take those timestamps and line them up with process metrics to know if something is driving a change for the better or for the worse. We also have a troubleshooting issue type. This is similar to an experiment, but it just represents a collaborative effort to diagnose or fix something. And we also have a hot pot issue, which is simply an agenda item that can be brought up for discussion in a larger weekly group meeting. So we're using JIRA here to build agendas for these large group meetings. We can pull up that agenda in the meeting. We also send out that agenda as a subscription the night before that meeting. So an example of the custom workflows we use here. So for an experiment issue, we've simply modeled this on the scientific method that you've all learned in 5th grade. So you have a hypothesis.
We did it in 4th grade in Canada, but that's all right.
Smarter up there. So you perturb the system in some way and observe what changes. You then gather the results and report your findings. So this is what that actual workflow looks like. I mean, it's a little bit more complicated, but the main pieces are in place. And these experiments can also then dovetail with the LCU sets project that John described. So some of these experiments that we do require sequencing on those instruments. So we can jump them off using a create and link and queue them up to go on the sequencer. So we're using Valiantis's Exoset plugin to do this. This is the new LNIO to manage these create and links. We're also making strong use of custom fields here as well. So if you're familiar with scientific journal articles, we've tried to create screens and fields that mimic the flow of a published paper. This makes paper writing, another activity that we're engaged in easy. I mean, by the time you're done with your experiment in JIRA, the paper has essentially written itself. So the last piece of the puzzle here involves grouping things logically so people can find the information that they're interested in. So we make use of JIRA's versioning features here. So John described a single sequencing process, but in reality, we're engaged in numerous processes centered around technologies or products that we're offering to our collaborators. So there might be a version 1.0 Illumina process that feeds these Illumina sequencers, and a version 3.0 Ion process that feeds these other sequencers, the Ion torrents. As the technology evolves, we can version that process and offer our customers additional features or efficiencies. And we would just make use of the fixed version to help us gather all the tasks and experiments related to releasing the next version here. We also make use of components in JIRA to logically group functional areas of that process. So Illumina and Ion might have a shearing component, both at their front ends. And folks working on shearing in either of those processes can then gather up issues that might be interesting to them. There are also functional groups that span these areas. So for example, John and his automation team, they may have interests and responsibilities for providing automation support across these different process areas. And then there are other sort of examples of these spanning functional groups. And we also make use of labels, but we sort of leave it up to the local users, the local teams to define how they want to use those. Okay, so what does this look like in the wild? So shown here is an example experiment issue. We have the custom fields here. We're just making use of the big text fields, and we're looking forward to the inline editor. This is going to make our job better. But I can describe what that experiment looks like here. Right, and this is sort of looking like that scientific paper. I can drop in charts for results. I can also have a conclusion sort of task. What turned out here? What's the next things that we need to go off and work on? I can also then scroll down and find out where this particular experiment lives in this hierarchy, so I can find its parent story issue or parent experiment that it derived from. Then we also have a lot of discussion going on in the comments area. I can also just take a peek in here to see that workflow that I just showed, just to see what steps are sort of available next. So this one's waiting for someone's manager there to approve it. And then here are some of the grouping functions using labels or the components to gather things up. So it's great, right? I can just click on one of these labels, and then that's going to take me to all the other related experiments for that project. I can find that parent task there. This is, I think, this is actually a troubleshooting effort, so there's lots of other experiments. We're going off gathering data, and I can see all the related tasks right next to each other. So, and then rolling it all up to get a view of this at the project management level, I can use Tableau then in a simple SQL query to pull in all the issues and those relationships. So what I'm showing here is just a Gantt chart. Again, based off of Tableau, I'm defining the lengths of those bars using the start and due dates from the database. Right now, this is all these issues belong to the LC component, the library construction component. And I can quickly use Tableau to filter this by other components, issue types, users, their states. And I can also build a table of all the other sort of related experiments. But there's also interactivity here. I can click on that story, and that'll instantly open up a browser where I can go right to that story issue, edit it, view it, work on it from there. So I'd say we're using JIRA in sort of a lightweight way for project management. And what I mean by that, it's not dogmatic. It's not like JIRA is taking over our own particular style of project management. We can sort of bend it to our culture in that way. And we like JIRA for that reason. So JIRA is flexible enough to mirror our particular style of project management. I just want to end by mentioning user adoption. So this has been a problem, or a concern anyways, for us when we've tried to launch other collaborative tools in our lab. And with JIRA now, we're seeing an increasing and sort of sustainable activity across our teams. So something we haven't seen with these other solutions. So here's activity in this Dev JIRA project, this eLab notebook project, plotted over the past year and a half. Again, I'm using Tableau just to pull those stats. And so this is all the activity, all the issues, stories, experiments, and comments on those issues over that time frame. I'm going to break that out by issue type, and you can see that we have over 600 experiments now in there, and there are nearly 60 ideas that could turn into other sort of potential products. So this is great. They're all cross-linked, searchable, and anyone in the organization can then learn about what other people are doing. So now I've just broken this out by user. So we're seeing a broad use across all the different users. And there's around 100 different users in this particular project. And some are using it more than others. But you can see we have some super users that are helping to pull others along. We can even sort of drill down and look at the bottom feeders here and maybe find out why they're not contributing that much. Let's see if I found, oh, John Stalker. John, what is that, one idea? What was the one bright idea you had?
Abstract for this talk?
That's a good idea. Thanks for, thanks for sharing. Let's get our people back here. We'll focus on one of our high flyers here. So this is Tony. She left our lab a couple of months ago, and her line ends here. But she actually did her entire master's thesis in our Dev JIRA project. She actually did it with like 187 different comments on one single issue. But encouragingly, she just sent me an email last week asking if we could stop over to her new lab and replicate this Dev JIRA, this eLab notebook solution for her lab. So that was really nice to hear, and I think we're looking to expand the use of this through different labs at the Broad, but also try and expand it out to other labs, get it out there, maybe use that marketplace to share some of these workplaces so other people that are doing this kind of work can sort of share. And I think that the keys to our success here with using this in this way is that JIRA just, it works. It makes people's jobs easier. easier. So organizing work by project and having historical records at their fingertips, it makes it an easy sell to most people. There's also user input here. So we take pride in being able to incorporate feedback from our users to tweak workflows and screens. And our users are really partners in its development. And also stickiness. So everything we do is in JIRA. So the day-to-day work that John described of tracking samples through the lab, that's done in JIRA. And then we just get the added benefits from being able to gather up ideas and track people's experiments in there as well. So I'd say as I just roam around the lab, it's probably like the second most popular website that I see up after Facebook. So again, if you could describe a business process, even genome sequencing, you can model it in JIRA. And with that, I think I'll close. And just thank you again. Thank you for all sticking around. Thanks, Ken. I mean, this has been a great concert. Feels like a concert. Conference. We don't go to a lot of software things, but everything I heard here was relevant. It's cool. You guys did a tremendous job. So thanks, and thanks to all of you. Just also want to mention a couple of our folks are here. So John, thanks. Amy Biasello is also here. I mean, she administers a lot of our projects, designs a lot of the workflows. John Walsh, who's not here, builds a lot of plugins for us. It also does a lot of integration with our robots. And then again, all of our users who help us build this. So,
and thanks wives, thanks Carrie for letting me come here.
Yes, thank you, Lisa.
Thanks, guys. Great job. Yeah.